Alright, जी अस्सलाम वालेकुम everybody. How are you all? Welcome back to my YouTube channel Biology by HR. So today we are going to be continuing question number 13, 14 and so on of the topical MCQs of AS level biology 9700 of mitotic cell cycle, right? हमने जो है we did the first 12 questions in the previous video and now I am going to do the last 12 questions with you guys starting from question number 13 over here. So let's dive into the class. Alright G, so now let's move on to question number 13 over here. Question number 13 over here is from May June 19, paper 1 to question 17. The question over here says that the diagram shows some of the stages which take place during the cell cycle. Right, cell cycle में हमारे पास 3 phases आ जाते हैं. One is the interphase, the next is the mitosis and the other is the cytokinesis, right? Now they have given us 1, 2, 3, 4 stages during the cell cycle. Now they say that which stages take place during the interphase. अच्छा, interphase specifically अगर हम बात करें तो interphase में we have G1 phase, we have S phase and we have the G2 phase. We know that the number of DNA molecule in the cell doubles in the S phase, which is the part of the interphase. So we can say yes, 1 is a part of the interphase. Now the cell growth. अच्छा, cell growth happens in the G1 phase of the cell cycle, where the you can say that where the cell increases in size and the organelles and the proteins are synthesized, right? So the growth of the cell occurs. जैसे कि cell में RNA synthesis होता है, organelle synthesis होता है, enzyme or you can say protein synthesis होता है which is required for the growth, right? So यहाँ पे हमारे पास cell growth का ये मतलब है, right? And it is happening in the G1 phase of the cell cycle. Chromosomes become visible. This happens in the prophase, right? This is not a part, and prophase is a part of the mitosis, not the interphase. Then it says that nuclear envelope disappears. Nuclear envelope also disappears during the prophase, right? So the correct answer over here would be 1 and 2. Why? Because they are asking the two stages that take place during the interphase. In interphase में 1 and 2 है. So the correct answer over here would be option A, 1 and 2 only. I hope that this MCQ is clear to all of you. Alright, so now let's move on to question number 14 over here, which is from February-March 2019, paper 1 to question 20. The question says that the embryonic stem cells are able to replicate continuously, okay? Then further in the question they are asking that what happens to the telomeres during the repeated mitotic cell cycles of the embryonic stem cells. अच्छा, we know that telomeres के बारे में what I told you guys that they are the non-coding parts, non-coding DNA sequences, right, which are found at the end of the sister chromatids. Sequences which are found at the end of the sister chromatids. और इनकी importance हमारे पास क्या होती है? They are important because they prevent the loss of the vital genes or the vital information during the DNA replication. During the DNA replication जब DNA polymerase copy करता है DNA को, so it does not copy the DNA till the end. So if there will be no telomeres, तो यहाँ नीचे से कुछ genes जो है हमारे पास जो है वो क्या हो जाएंगी, चली जाएंगी और delete हो जाएंगी, ठीक है. So because of the telomere, what happened that some bases from the telomeres are lost, not from the DNA, right? और DNA का नॉन कोडिंग पार्ट क्योंकि DNA का जो नॉन कोडिंग सीक्वेंस है वो क्योंकि डिलीट हुआ है, बिकॉज टीलोमेयर इज द नॉन कोडिंग सीक्वेंस ऑफ द DNA, वो लॉस्ट हुआ है. सो एज अ रिजल्ट टीलोमेयर शॉर्टन्स, ठीक है. सो विद द रिपीटेड माइटोटिक सेल साइकिल द टीलोमेयर्स shorten, right? So hence the correct answer over here would be their length decreases. So hence the correct answer over here is option B. Obviously option A cannot be the correct answer. Their length increases, no, because with the with every cell division the telomere length shortens, right? They are completely lost, no. They stay at the same length, no, right? So hence the correct answer over here would be option B. So the correct answer over here would be option B. I hope that this MCQ is clear to all of you. अगर आप लोगों को और इसमें डिटेल चाहिए so I can give it, कि जैसे कि ये हमारे पास एक DNA का मॉलिक्यूल है, राइट? दिस इज़ द DNA मॉलिक्यूल और ये हमारे पास टीलोमेयर है. obviously the DNA polymerase is not copying the DNA full, right? So what happens is that, for example, उसने इतना copy किया, so the telomeres are lost with the subsequent cell division, not the coding DNA. ये इसकी importance होती है. I hope that you have got it. Now let's move on to the next question. Let's move on to question number 15 over here. Question number 15 is from October November 18 paper 1 question 19. The question says that how many copies of each DNA molecule will be found in a cell at each stage of the mitotic cell cycle. We know there is the G1 phase, there is the S phase, then there is the G2 phase, which is the interphase, right? And then there comes the mitosis, and then there comes the cytokinesis, right? Basically, the DNA is replicated during the S phase of the cell cycle. So during the G1 phase of the interphase, there would be one DNA molecule only, right? In the cytokinesis, we know in the cytokinesis, the cell is something like this, and the contractile ring is formed between the two nuclei, right? The contractile ring contracts and the opposite membrane fuse, and then the two daughter cells are made. But after the cytokinesis, the daughter cells are made, right? After the cytokinesis, the daughter cells are made when the cytokinesis is completed. During the cytokinesis, the how many copies of the DNA molecules will be found in the cell which is undergoing cytokinesis? It will be 2, right? And then the contractile ring will be formed, cleavage furrow will be formed, the opposite membranes will fuse together, and the two daughter cells will be formed, right? So cytokinesis mein the cell is going to have 2 DNA molecules, hence the correct answer over here would be option B. I hope that this MCQ is clear to all of you. Let's move on to question number 16. Question number 16 is saying that which events listed are the part of the mitosis, interphase, prophase, cytokinesis. So I already told you guys that G1 से S phase तक, G1 से G2 phase तक we are having the interphase, right? So interphase is not a part of the mitosis. Obviously these all are the part of the cell cycle, right? These all are the part of the cell cycle, but not particularly the mitosis. Mitosis में कौन कौन से part आ जाते हैं? Prophase, metaphase, anaphase and telophase. So prophase, yes, prophase is a part of the mitosis. Cytokinesis is not a part of the mitosis. Cytokinesis भी हमारे पास अलग से आता है, right? So hence the correct answer over here would be option D, 2 only. Now again, a question number 17 is a similar question we did in question number 15. वो यही पूछ रहे हैं in question number 17, which is from October November 18 paper 1-3 question 20. They are asking that how many copies of each DNA molecule will be found in a cell at each stage of the mitotic cell cycle. In the G2 phase, how many DNA molecules will be found? Obviously G2 phase is coming after the S phase, and S phase में हमारे पास क्या होती है? DNA replication होती है, right? In the S phase we have the DNA replication. So हमारे पास जो है in the G2 phase there will be 2 DNA molecules. In the metaphase, obviously metaphase में क्या होता है? That the chromosomes are aligned at the center or the equator of the cell, and metaphase is a part of the mitosis. So over there we will find, over there also we will find 2 DNA molecules, right? 2 copies of each DNA molecule. Cytokinesis may also there will be the 2 DNA molecules, hence the correct answer over here would be option D. I hope that this MCQ is clear. Now let's move on to question number 18. Now question number 18 is from May-June 18 paper 1 question 21. It says that which statement is correct about the number of telomeres present in the prophase of the human body cell. Prophase, in the prophase we know that the DNA has already been replicated, right? DNA has already been replicated in the prophase of the cell cycle. So what will happen, that there are 46 chromosomes, right? In the human cell there are 46 chromosomes, and each chromosomes consist of 2 sister chromatids. Each chromosomes consist of 2 sister chromatids. This is one chromosome which is consisting of 2 sister chromatids, and the telomeres are found at the end of the each sister chromatid. So we can say that one chromosome one chromosome is having 4 telomeres, right? 4 telomeres. So how many telomeres will the 46 chromosomes have, right? So 46 chromosomes will have 184 telomeres, right? 46 into 2 into 4 is 184, right? So there would be 184 telomeres, as there is one telomere at the end of each 92 chromatids. Why they are saying 92 chromatids? Obviously one chromosome is having two sister chromatids, so 46 chromosomes will be having 92 chromatids, right? So hence the correct answer over here would be option D. I hope that this is clear. Now let's move on to question number 19. Alright, so now let's move on to question number 19 over here, which is from October-November 17, paper 1-1, question 17. The question says that colchicine is a chemical that stops the chromatids from separating during the mitosis. A cell is treated with colchicine, right? Which stage of the mitosis will this cell reach but not complete? Now, what is the chemical doing? Chemical is basically stopping the sister chromatids from separating during the mitosis, right? In which phase of these mitosis does the sister chromatid separate? It is the anaphase, right? What happens in the anaphase? That the centromere split and the sister chromatid, the spindle fibers shorten, and the sister chromatids separate and move towards the opposite poles of the cell. So obviously it is not going to reach the anaphase. It is definitely going to reach the metaphase, right, where the chromosomes align at the center of the equator. But as the chromatids cannot separate, and therefore the cell cannot enter the anaphase, and so what will happen, that the cell will get stuck in the metaphase, right, and anaphase will never begin. So hence the correct answer over here would be option B, metaphase, right? Alright, so moving on with question number 20 over here, which is from February-March 2017, paper 1 to question 22. The question says that the enzyme telomerase repairs the telomeres. It stops the telomeres from getting shorter each time a chromosome is replicated. We know that with each cell division, the telomeres shorten. But with the presence of the telomerase, what happens is that telomerase is that enzyme which will add that DNA repeats back to the telomeres, which will prevent the telomeres from shortening, right? So आगे ये लोग कह रहे हैं that telomerase is not normally active in the human body cells, but in some diseases telomerase can be activated. अच्छा, बिकॉज यूजली हमारी जो बॉडी सेल्स होते हैं उसमें टीलोमरेज इज यूजली इनएक्टिव. सो आफ्टर सर्टन नंबर ऑफ डिविजन्स टीलोमेयर्स इतने ज़्यादा शॉर्ट हो जाते हैं बिकॉज दे आर बिकमिंग शॉर्ट आफ्टर ईच सेल डिवीजन, राइट. और टीलोमरेज भी हमारे पास इनएक्टिव होता है हमारी बॉडी सेल्स के अंदर. सो व्हाट हैपेंस इज दैट आफ्टर अ सर्टेन नंबर ऑफ सेल डिवीजन टीलोमेयर्स इतने ज़्यादा शॉर्ट हो जाते हैं दैट द सेल स्टॉप डिवाइडिंग और द सेल डाइज, राइट, जो कि प्रोग्राम सेल डेथ होती है, एपोप्टोसिस होता है, राइट. सो यहाँ पे कह रहे हैं कि टीलोमरेज जो है वो नॉर्मली हमारी बॉडी में इनएक्टिव रहता है, बट ड्यूरिंग सम डिजीजेज हमारे पास टीलोमरेज एक्टिवेट हो जाता है, राइट. देन दे आर सेइंग दैट इन विच डिजीज इज द एंजाइम टीलोमरेज एक्टिवेटेड. अच्छा, अगर हमारे पास टीलोमरेज एक्टिवेट हो जाएगा सो द टीलोमेयर्स वोंट शॉर्टन, टीलोमेयर्स won't shorten, right, and if the telomeres won't shorten, so cell will subsequently divide. Cell will subsequently divide and it will not stop its cell division. So cell will subsequently divide and it will keep dividing, which will contribute to the tumor ग्रोथ, राइट. जो ट्यूमर होता है जो कि हमारे पास कैंसर्स में होता है. सो बेसिकली टीलोमरेज अगर हमारे पास है तो इट इज़ एक्टिवेटेड मोस्टली इन द कैंसर, ठीक है. वाई? बिकॉज हमारे पास कैंसर में क्या हो रहा होता है दैट देयर इज़ द सब्सिक्वेंट सेल डिवीजन एंड द सेल कीप्स ऑन डिवाइडिंग, जो कि हमारे पास ट्यूमर ग्रोथ कर देता है, राइट. सो हमारे पास जो सेल है वो सब डिवाइड हो जा रहा है, इट इज़ नॉट स्टॉपिंग. तो उसका यही मतलब है दैट द टीलोमरेज इज़ एक्टिवेटेड. अगर इनएक्टिवेटेड होता है तो आफ्टर सम सेल डिविजन्स जो है वो टीलोमेयर शॉर्टन हो जाते हैं, फिनिश हो जाते हैं और सेल वुड स्टॉप डिवाइडिंग. ये ह्यूमन मैकेनिज्म है, ह्यूमन सेल मैकेनिज्म है ये, ठीक है. सो आई होप दैट दिस इज़ क्लियर टू ऑल ऑफ यू. नाउ लेट्स मूव ऑन टू क्वेश्चन 22 विच इज़ फ्रॉम October-November 17, पेपर 1-3, क्वेश्चन 20. The question over here says that what describes the telomere. सो व्हाट इज़ अ टीलोमेयर, हमने अपने जो लेक्चर नोट्स हैं, लेट्स रेफर टू द लेक्चर नोट्स. टीलोमेयर्स हमारे पास क्या होते हैं? दे आर द ग्रुप ऑफ नॉन कोडिंग मल्टीपल रिपीट सीक्वेंसेज ऑफ द डीएनए फाउंड एट द एंड ऑफ द ईच क्रोमाटिड. Right, so what are the multiple repeat sequence? It is the region of the repeated nucleotide sequences at the end of the each sister chromatid. So hence the correct answer over here would be option D. The telomeres are the region of the repetitive nucleotide sequences which are non-coding. They do not code for a specific protein, right? At the end of the each sister chromatids. They are found at the end of the each sister chromatids. अब तो मुझे लग रहा है आप लोगों को ये पूरा याद हो गया होगा because we have discussed it so many times, right? Alright, so now let's move on to question 23 over here, which is from October-November 2018, paper 1 to question 22. The question over here says that which row shows some of the events during the mitotic cell cycle? Which row shows some of the events during the mitotic cell cycle in the correct sequence? Right, so let's read the options one by one and try to get our correct answer. Centromeres of each chromosome splits. Nucleoli becomes visible. Nuclear envelope breaks into small vesicles. अच्छा, basically जो centromeres होते हैं of the each chromosomes, it splits in the anaphase, right, and the sister chromatids move apart. न्यूक्लियोलाई बिकम विजिबल अगेन ड्यूरिंग द टीलोफेज, बट ये जो हमारे पास इसके आगे लिखा हुआ है दैट द न्यूक्लियर एनवेलप ब्रेक्स इनटू स्मॉल वेसिकल्स, सो दिस इज नॉट द केस इन द आफ्टर द टीलोफेज. इट इज़ नॉट इन द सीक्वेंस, इट हैपन्स इन द प्रोफेज. प्रोफेज में हमने पढ़ा है दैट द न्यूक्लियर एनवेलप डिसइंटीग्रेट्स इनटू द स्मॉल पीसेस, राइट. सो यहाँ पे दिस डज नॉट फिट्स इन, सो ऑप्शन ए हम लोग एलिमिनेट कर देंगे. Let's have a look at option C. Nucleoli becomes visible. No. Nucleolus या फिर nucleoli जो होता है it disappears in the prophase. Disappears in the prophase. पहले वो disappear होगा and then it will reappear again. Nuclear envelope breaks into small pieces. ये सही है, but this is not correct. Nucleoli becomes visible. So hence this option will also be eliminated. Then it says that microtubules are produced. Centromere of each chromosome splits. Nucleoli becomes visible. यहाँ तक सब कुछ सही है, ठीक है, कि spindle microtubules are produced. They are going to attach to the kinetochores in the centromere of the chromosome, and the sister chromatid is going to split. Nucleoli then will become visible in the telophase. But over here it is written that the nuclear envelope breaks into small vesicles. No, जब nucleoli हमारे पास visible होना शुरू हो जाएगा in the telophase, so the nuclear envelope will also begin to reform in the around the each chromatid. Around the nuclear envelope will also reform in the telophase, right? But over here they have written that the nuclear envelope breaks into the small vesicle which happens in the prophase. So this is not in the timely order. हमें timely order में बताना है. So the correct answer over here would be option B, क्योंकि यही बचा है हमारे पास. So the correct answer over here would be option B. Let's read the option. Nuclear envelope breaks into small vesicles, जो कि हमारे पास प्रोफेज में होता है. स्पिंडल माइक्रोट्यूब्यूल्स प्रोड्यूस होते हैं उसके बाद, एंड देन द centromere of the each chromosome splits in the anaphase and the nucleoli becomes visible in the telophase. This is giving the whole correct sequence, right? So option B is the correct answer over here. Now let's move on to question number 24 over here. Now let's move on to question number 24 over here. Question number 24 is from October-November 2017, paper 1 to question 21. The question says that cancer cells may divide by far more divisions than other cells found in the humans. Which statement about the cancer cells is correct? So basically हमने cancer cells के बारे में अभी in the previous some questions हमने ये चीज़ स्टडी की है that they are able to synthesize the enzyme telomerase, and the enzyme telomerase is activated in the cancer cells, right? So if the enzyme telomerase is activated, which means that the cell is going to divide multiple times and the cell division will not stop and it will form a tumor, right? अगर हमारे पास telomerase नहीं होता, so the cell division would have stopped after some time. But in the cancer cells कैंसर सेल्स में टीलोमरेज एंजाइम्स एक्टिवेट होते हैं. ऑल्सो दे आर एबल टू, जो कैंसर सेल्स होते हैं, दे आर एबल टू सिंथेसाइज द एंजाइम टीलोमरेज. If I read the other options, they have a mutation in the telomeres, so the DNA is not hydrolyzed. No, there is nothing like this happening. They have DNA polymerase who can replicate their DNA without the telomere loss. No, DNA polymerase जो होता है, it does not replicate the whole DNA. ठीक है, it does not replicate the whole DNA, तो DNA loss तो ज़रूर होगा, right? So this is also incorrect. They increase the number of copies of the repeated DNA sequences in the telomeres. No, increase नहीं करते, it remains the same, right? मतलब कि वो shorten नहीं होते telomeres जो होते हैं वहाँ पे. So I hope that this MCQ is clear to all of you. The correct answer is option A. So I hope that you found this video of topical MCQs of AS level biology 9700 mitotic cell cycle very much helpful. If you guys did, do consider subscribing this channel and do share this video with your friends as well, so they can benefit from it as well. Do remember me in your prayers. Do send good prayers on my way. And that is it. Good luck for your upcoming examinations. Do prepare well for your AS level biology. Take good care of yourself. Allah Hafiz everyone. See you guys in the next video.